I would now like to invite Dr. Rae Yeung to discuss her work on a little bit of switch in topics on immunology in terms of Kawasaki disease. Dr. Rae Yeung is a professor of pediatrics, immunology, and medical science at the University of Toronto, senior scientist in cell biology research, and the Hackman and Deborah Chu Chair in Pediatric Translational Research at the Hospital for Sick Children. Dr. Yeung's research focus is to understand the mechanisms governing autoimmunity, specifically the mechanisms involved in initiating and sustaining the immune response in childhood arthritis and rheumatic disease. Dr. Yeung.
So good morning, everybody. It's great to be here to see all the excited faces in the audience. It's pretty difficult, I think, to learn all about the different spectrum that you're learning about. So I'm just going to take you through a little journey, and forgive me if I speak a little quickly because I get very excited about what I do. So wave or something if I'm speaking a little too quickly. I'm a pediatrician. I work at Sick Kids, and I look after children with a disease called Kawasaki disease. So I'm going to take you through a little bit from the clinical perspective, what is Kawasaki disease, then talk a little bit about some research that we do in the lab, and then see how the two can actually merge together. And much like what Dr. Mount just talked about, how the mice and the men actually really work together to help us improve treatment and outcome for affected children. So what is Kawasaki disease? So everybody, there's the requisite gross picture of what a disease looks like. These are disease that affects young children, usually children under the age of 5, and they develop fever, prolonged fever, inflammation, which affects basically most organ systems in the body, including the skin, leading to a polymorphous skin rash, inflammation of the eyes, leading to these red eyes, which are not pussy, the inflammation of the oral mucosa, which leads to red lips and cracked and a strawberry tongue, inflammation of their lymph nodes, as well as swelling of the periphery of their hands and their feet, with swelling of the dorsum of the hands and redness of the palms and soles of the feet. And this very, very unique syndrome actually affects the blood vessels, which are supplying all of these organs. And was initially described for this guy in the middle of the picture named Dr. Kawasaki. Actually, is this a pointer? Is this a pointer? So this guy here with the sombrero, he's not Mexican, he's actually Japanese. And he's a pediatrician who actually in the 1960s recognized and described this illness and called it mucocutaneous lymph node syndrome, really describing this constellation of inflammation affecting the eyes, the lips, the skin, and all of that. And what he found was that not only these kids developed all this redness, the classic signs of inflammation, heat, swelling, and redness, they actually developed inflammation of a very important blood vessel in the body, and that blood vessel is the coronary arteries. So we all know that the coronary arteries actually supply blood to the heart, and in children with Kawasaki disease, they develop inflammation and breakdown of the coronary artery wall, leading to aneurysm formation. And this is really an angiogram looking at a giant aneurysm in a child with Kawasaki disease. So these are young children that are now running around with basically aneurysms in their heart. And I had the pleasure of meeting actually the index case, the very first one that was described in 1967 with Kawasaki disease. And he's a 50-something-year-old now who has had multiple heart attacks because of these large aneurysms which disrupt blood flow to the heart. So there are many lessons that we can learn about Kawasaki disease. We can learn lessons from the epidemiology, meaning what is the pattern of disease as it affects, and how do we see this in affected children? Well, what we do know about Kawasaki disease, it is actually much more common than we may believe. You may never have heard of it, but I'm fortunate to see many of them at Sick Kids, and I see between 100 and 150 new cases of Kawasaki disease per year. And over the last about umpteen years, we follow now over 2,300 kids at Sick Kids with Kawasaki disease. It affects boys a little bit more than girls, so that the sex ratio is about 1.6 to 1. And as you can see, looking at the age of distribution, it's really very young children that develop this disease, children under the age of 5. In fact, the peak age of incidence is about 18 months. What can we learn about the epidemiology is that no matter where you look in the world, whether it's Canada or much more prevalently in Japan, the spectrum of age distribution and seasonality is the same. When you look at a disease that really is a little bit rare under the age of 6 months, one of the things that it points to is perhaps this is something that there is maternal protection conferred to individuals. So you have to kind of think about what is it that's rare and what is it that can be protecting you, what that's acquired in utero. And what is it that after the age of 5 it becomes exceedingly rare. And most of these things, that is maternal protection and then rareness after the age of 5, points to really something that the immune system learns to see. We all acquire passive immunity from our parents, from our mother, and this usually protects you from common infectious diseases in the early neonatal period. And then you usually are exposed to common pathogens before the age of 5, so that after the age of 5, your body, your immune system has actually learned to basically protect and have good memory to confer protection against infectious diseases. The other evidence that points to an infectious trigger for Kawasaki disease is really the pattern of disease over years, so that we know that this is an endemic disease with sporadic epidemics. So the blue is boys, the red is girls, and you can see that there are epidemic years as long as we've kept track of how many cases of Kawasaki disease we've seen. And this is seen both in North America as well as in Asia, and the epidemic years are the same years, much like influenza. So the fact that the age distribution, the years distribution, and the seasonality really points to that there's potentially an infectious trigger that's leading to this disease. And Kawasaki disease is often preceded by a snuffly nose or diarrhea, and kids and their siblings usually get a case of Kawasaki disease within 10 days of the index case, really pointing to the search for an infectious trigger. So many people, everybody who's been in Kawasaki disease research have actually looked for infectious triggers. And you can see that it's a very long list of both viruses and bacteria that have been implicated in Kawasaki disease. But what's really interesting that there hasn't been just one organism that's been found. So we've needed to look for something that may be common to these microorganisms or something that may be common to the responding host. So the first thing that I'm going to just take you through a little bit is actually what may be common in the genetic host. And what is common in the host may be the role of genetics, how we're made, how we're actually put together, and our immune responses. And we know from worldwide surveillance that the incidence of Kawasaki disease varies tremendously across the world, from the lowest in Denmark and New Zealand, single digits, to the highest in Japan. In fact, the most recent surveillance in Japan shows that the incidence is about 230 per 100,000 children under the age of 5. And in fact, when children in Japan move to the United States, their incidence does not change. So they did pointing to really the strong role of genetics. So when the kids moved from Japan to Hawaii, they retained the incidence of Kawasaki disease for Japanese children and don't kind of take on the environmental incidence of what children in the United States would, really pointing to the important role of genetics. So in collaboration with the International Kawasaki Disease Genetics Consortium, we have actually been looking at key pathways using both candidate gene approach as well as genome-wide association studies to look at what may be important in our genome that's leading to predisposition to Kawasaki disease. And today I'm going to focus a little bit and just give you a flavor of what we've looked at in terms of immune activation. Now, one of the things about studying a disease, much like what Dr. Mount was talking about with Alzheimer's disease, is it's very difficult to actually say, can I have a little piece of your heart so I can study what's happening in your heart? Much like you can't do that for brains in Alzheimer's disease, so we rely on an animal model. So animals, mice in this case, give their lives willingly to science. You know, we take their hearts whenever we want, and we give them Kawasaki disease. And one of the lessons that I learned very early on in science is that many things happen serendipitously. So when you least expect it, a discovery is made. And such was the case for Kawasaki disease, the animal model of Kawasaki disease. So Tom Lehman, who was a rheumatologist from New York, was actually trying to look and develop a model of reactive arthritis. And he ground up Lactobacillus cell wall because it looked very much like streptococcal cell wall, thinking of inducing a reactive arthritis model. But when he injected this Lactobacillus, which is the stuff you eat in yogurt, into the mice, they developed coronary inflammation instead, blood vessel inflammation instead of joint inflammation. And he then went on to characterize this model and called it the model of Kawasaki disease, because what happens is when you inject the mice with this lactobacillus, a cell wall extract, they start to develop inflammation around the coronary artery. So this is their coronary artery, this is the aorta. The base of the aorta is where the takeoff of the coronary arteries are. And by about day 28, there's a mononuclear cell infiltrate, which really kind of surrounds the coronary artery and then leads to elastin breakdown, which you can see using an elastin von Giesen stain. This stains the elastin layers in the arteries, and breakdown of elastin is really the hallmark of aneurysm formation. So these mice, much like young children, only young mice get this disease, and young mice break down the elastin in their coronary arteries and develop aneurysms, much like Kawasaki disease. We went on to characterize a common feature in many microorganisms called the superantigen, and I won't go into gory detail, but when we found a novel superantigen within this yogurt bacteria, Lactobacillus, which actually causes Kawasaki disease, it initiates a massive immune response when presented to T cells, and T cells can only do several things. They can make many more of themselves, and they can make many soluble mediators of inflammation. One of which is TNF. And I'm going to focus the next minute on talking a little bit about TNF. And TNF, or tumor necrosis factors, is a very important pro-inflammatory mediator that leads to many, many different downstream effects. One of the very important downstream effects, and TNF is a very important molecule because it's been found to be important in many inflammatory diseases that rheumatologists look after, including arthritis and vasculitis, or inflammation of the blood vessels. So the first thing we did was we looked to see if these mice, when they were developing Kawasaki disease, did they have TNF in their hearts? And sure enough, they made TNF at the message level. And if you see here, the red dots are TNF basically being made in the coronary artery. This is the isotype control, and this is the saline injected negative control. And in fact, TNF can do many, many things. But one of the critical things that we found TNF did is it actually mediates production of a very important matrix-degrading enzyme called MMP9, or matrix metalloproteinase 9. And MMP9 breaks down elastin. Remember, I showed you the picture of the coronary arteries breaking down their elastin. So when you actually have mice that have been given the extract to cause Kawasaki disease, we show that they actually are making a lot of MMP9, in this case lighting up in red again. And when we block TNF with a drug that's in the clinic called etanercept, or Enbrel, we can actually make that go away. And when we use knockout mice that do not have a TNF receptor, they again do not have any production of this matrix-degrading enzyme. And when we used colocalization, we saw that actually the smooth muscle cells here lit up in green, and MMP9 lit up in red. And when you put the two together, you get yellow. It is actually the smooth muscle cells, which is within the coronary arteries of these mice that are developing Kawasaki disease, which are actually making this enzyme, which is breaking down elastin. And when you put a little layer of gelatin on a slide with the heart on it, and the slide is infused with something that glows when the elastin breaks down, what we see here it's glowing, and here's the coronary artery and the aorta, that actually this enzyme is actually active. So it's biologically active, and it's breaking down the elastin, which is around this coronary artery. But when you look elsewhere in the heart, it's actually not breaking down anything else. So there's something very unique about the environment and the specificity, organ specificity, of why it targets the inflammation and the breakdown of the coronary artery around the heart. So to make a long story short, we started really going into this story and looking at what's happening downstream, and we found that all these little pieces of elastin were actually being broken down into little pieces. And these little pieces of elastin, or elastin-derived peptides, can actually be measured in the circulation of mice that are developing aneurysms, not the ones that don't have the MMP9, but the wild type who do, and children with Kawasaki disease versus febrile control, so other children that have fever but not Kawasaki disease. So when you break down your coronary arteries, you can actually release these little pieces of elastin. And the fascinating thing was these little pieces of elastin were not inert. they were actually biologically active. So that when you sprinkle these little pieces of elastin on smooth muscle cells, they made more of these enzymes that can break down elastin. So these elastolytic MMPs, which is MMP 9, 3, 12, and 2, they actually upregulated at the transcriptional level how much they made. And in fact, the nice thing about being in Toronto is there are experts everywhere. And I'm not an elastin expert, okay? But right down the street from me was actually somebody, Alec Hinnick, who actually cloned the elastin-binding protein. So he knew that there was a protein, which is a splice variant of beta-galactosidase, and this is a lysosomal enzyme that binds galactose sugars, actually binds and signals elastin protein. And when galactose sugars, such as galactose or lactose, binds it, there's a conformational change, and this receptor is shed from its surface, so signaling via the receptor is inhibited. So what does that mean? That means that perhaps we can use galactose sugars as a therapeutic agent to inhibit signaling of this particular elastin and break this vicious cycle. So when we sprinkled lactose into these smooth muscle cells, sure enough, they actually downregulated the transcription or production of elastolytic enzymes, and in fact, we can inhibit signaling, which is actually via the MAP-ERK pathway. So to really bring this full circle, we just need to ask ourselves, what are the implications in children? So if you believe what I just told you, that inflammation with a common property of infectious triggers, which is a superantigen, leads to activation of T cells. T cells migrate to the coronary artery. Then they tell the TNF, they make lots of TNF, and they tell the coronary artery to make lots of TNF. And then the TNF actually drives upregulation and production of this matrix-degrading enzyme by the smooth muscle cells. And when they break down elastin, these little pieces of elastin-derived peptides are actually active. And it forms a positive, vicious feedback cycle to make more MMPs and turn on the immune system even more. So we're now going to try to turn this off. And one of the things, and we know we can turn it off with lactose. So what does it have to do with Kawasaki disease and genetics? Well, I want to show you a heat map of Kawasaki disease on the top and the incidence of lactose intolerance on the bottom. So who doesn't handle lactose well, actually? It's the people who are actually most susceptible to Kawasaki disease. And although it may be kind of silly to suggest, but could it be as simple as drinking a glass of milk that can turn off your immune response to actually shut down elastin breakdown? And this is actually what we're doing in the mice right now, is that we're feeding them lactose to see if we can turn off the elastin response. So in the interest of time, I'm actually just not going to talk about, other funny things, but I did want to talk about one thing that I think was is to bring back science to really the level of where I started when I sat in your seats, and what we're doing right now. I hope I've been able to show you that we can actually take human data that we learn about genes, go into the mouse, ask about RNA, ask about proteins, ask about what they do in cells, ask about what they do in tissues, and then combine that with some of the infectious and environmental triggers that I didn't get a chance to talk about yet, and see how they actually converge and act in patients, and hopefully be able to improve the outcome by using targeted therapy to many of the genes and molecules that we find in the molecular and cellular pathways. So that's kind of where we are. And what I wanted to bring it back to is, again, when I started in your seats, okay? Science is amazing, and I hope that you've heard all about a little flavor of neuroscience earlier, a little flavor about immunology here. How long, and you're going to hear more as the hour goes on. What do we have to do to get here? Well, for me, I wanted to just share my personal story, okay? Is that I did 2 years of undergrad. Back then, in the eon days, you could get into medical school after 2 years, so I did. I did 4 years of medical school, and then I did 4 years of pediatrics. I did a PhD afterwards, and I did subspecialized in pediatric rheumatology. Some of them overlapped, but it's 16 years in university, okay, guys? But it's unbelievably worthwhile, okay? Anything that is worthwhile is worth spending the time doing, okay? And I think has not been a year of my education that I've regretted, and I continue to learn every single year. And the benefits of being able to wake up every morning and feel like you want to go to school, want to go to work, is actually outweighs any of the time that you're going to spend in university. So although you may be in first, second, or third year right now, as you look forward, it may seem like forever. It's longer than high school if you think about it, right? You spent 12 years in elementary school and high school, spent a little longer than that in university, but it's unbelievably fulfilling, and you get to travel the world. So I formed a consortium. So these are all my colleagues in Canada. So all of the people in pediatric rheumatology, we all get together, and we're all doing research together, so I get to fly all over Canada and meet them. I formed an international consortium called Understanding Childhood Arthritis Network, and these are people all over the world who work together with me, and we were all dressed up because we were meeting the Queen of the Netherlands because she presented us a big check to do our research. So there are lots of benefits, not just to doing research, but to meeting lots of colleagues, and the journey is long, but the journey is very, very worthwhile. And I just wanted to end by thanking the colleagues and collaborators who actually do the work and the funding agencies that fund us. And a picture of the lab. So this is no slave labor here. These are just kids, okay, that are members of the family, okay? So, one of the nicest things about being a PI is you just get to dream the dream. And guys like you do the work, right? So these are the guys that do the work. And you get to actually have a family beyond just your biologic family, but your lab family. So very worthwhile journey, and I wish you all the best as you go on and decide what you do in your research futures. Thanks.